Welcome back. This is Dr. Vishal Trivedi from Department of Biosciences and Bioengineering. So, let us continue about our discussion about the sexual reproduction in flowering plant, and what we were discussing in the previous lecture is we have discussed about the male sex organ, and we have also discussed how the pollen grains are being formed in the male sex organ, and then we have also discussed about the dispersal of pollen grains from the one flower to another flower, and we have also floated several crucial questions, and I requested you in the previous lecture that we should attempt and understand these questions either by browsing the web sources or the literatures. So, we will start the lecture with the addressing these questions. So, let us see what are these questions are. Question 1. Okay. So, the first question was what is chasmogamous flower, okay, and the answer is that the chasmogamous flowers are the normal flower which are found in the environment, and chasmogamous flowers are the flower which are having the exposed anther and stigma. What does this mean is that most of these flowers actually will prefer to go for cross pollination, but as I said, there is no such rule that they will do the cross pollination, but in general, since they have the exposed anther and stigma, they probably will follow the cross pollination, and they are actually open for the any kind of the pollination mechanisms. The question 2 says the can cross pollination possible in a clistogamous flower. So, if you remember our previous lecture, we have also discussed about the clistogamous flower. So, clistogamous flowers are the flower which are where the flower is closed, and it is actually in the closed condition itself, the anther as well as the stigma are matured. So, in these kind of flowers, the cross pollination is not possible because the flower is closed. So, if the flower is closed, the external agent cannot come and do the cross pollination, and examples in this class is the oxalis. Next question is mention two strategies which are being evolved to prevent the self pollination, okay. And I think we have discussed about this. We have discussed what are the two strategies. We will discuss even more. So, there are multiple strategies. One of the strategy is, so strategy number 1 is called hercogamy. Hercogamy means a kind of mechanical barrier, okay. So, this mechanical barrier could be gynostegium, or pollinia, okay. So, in the case of gynostegium, the stigma part is covered with a membrane like structure, whereas in the case of pollinia, the anthers are also covered, and this kind of mechanical barrier will never allow the self pollination, and it will actually needs a particular agent, mostly the biotic agent, to remove these barriers or actually hinder penetrate the barriers and then facilitate the cross pollination. The next factor which is favoring towards the cross pollination or which is being evolved to prevent the self pollination is the dichogamy. Dichogamy means the development of anther and stigma on different time points. Okay. What is this mean is either the male sex organ is evolved, even if it is a bisexual plant, either the male is evolving, male is developing first or the female is developing first, and in these kind of situation the self pollination is not possible because the same anther, even if it is evolved, even if it falls onto the female organ or stigma, that will not going to work because the female sex organ is still not evolved. In this case we have two different conditions, one is the protogyny and the other one is called as the protoandry, and in the case of protogyny it is the development of female sex organ, whereas in the case of protoandry it is the development of the anther earlier than the gynoecium. Now, we have the fourth question, and the fourth question is as you can see on the PowerPoint slide, the fourth question is what is the mechanism of emasculation or what is emasculation in plants. So, question number 4, what is emasculation in plant? Okay. So, emasculation is actually a procedure to control or to avoid the self pollination in a artificial system. So, what we have discussed so far, we have discussed how the mechanisms are being evolved in the plant to avoid the self pollination, but this is the mechanism which actually is being artificially being done by the humans to not facilitate the self pollination or within the plant. So, you can imagine that you have a flower where you have, this is the stigma and anthers are coming like this. Okay. So, in these cases like this is the flower. Okay. Okay, so this is the corolla and this is the anther and this is your stigma. Okay. So, what is mean by the emasculation is that you kill the anther, and once you kill the anther, this plant is not going to get the self pollination from its own flower. Emasculation is also being done in the cases where the plants are unisexual, but that possibility is very low. Most of the time the emasculation is done by the bisexual flower to avoid the self pollination, and it is actually a part of the procedure in the horticulture technique to evolve the new species or the transgenic species or the more advanced techniques, more advanced species. And these kind of more advanced techniques are known as the bagging technique, and that actually is help in the breeding. So, that is our next question is what is the bagging technique and how it is useful in plant breeding program. So, bagging technique is that, what is bagging technique? The bagging technique is that, so you have this. So, first you do emasculation. So, what is mean by emasculation is that you have this flower, this is the stigma, and you have removed the top portion. Okay. And So, this is the emasculated flower. Okay. Now, what you do is you cover this flower with a polythene bag, and that is called as the bagging technique. How the bagging technique is useful in plant breeding. So, after that what you are supposed to do, this is actually a plant 1, okay, and actually you are trying to do the cross pollination in this plant rather than the facilitating the self pollination. So, what you have done, you have first done the emasculation, you have removed the anthers or you have destroyed the anther. So, now you have a plant which does not contain the anther, but you do not want a cross pollination to happen, because the anthers can come from a through cross pollination from the neighboring plant. So, what you have done, you have covered this whole flower in the form of bag, and this is mostly being done when the flower is still in a budding condition, means it is still in the bud. So, what you have done, you have removed the anthers, you have destroyed the anther when it was in the budding condition, and then you have covered the bud. So, after that it is actually being protected from the self pollination. So, by the emasculation you have destroyed the self pollination, by a bagging technique you have actually destroyed the cross pollination. Now, this flower is ready to receive the new pollen grain, okay, and that new pollen grain you can take up from the plant 2. So, how you can take up the new flower or new pollen grain? So, you remember that this is the anthers, okay. So, what you have to do is you take up the anther, okay. So, imagine that it is a having the multiple anthers, okay. So, what you do is take a paper, okay. So, take a paper or take a paper like this, okay, and keep your anther on top of this paper, okay, and then tap this anther, okay. So, once you tap this anther, the pollen grains will get collected here, okay, and then you can collect these pollen grains into a tube, and you can store them either. You can, so these are the called as appendophs. So, what you can do is you can just tap the anthers on a paper. So, the pollen grains will fall onto this clean paper. You can take this clean paper and transfer that into the appendoph. Now, your appendoph has the anther. Now, you can take these anthers and do, and once you can remove the polythene and you can add those to the stigma artificially, and that actually will allow to generate the new crop species or new crop plant. So, that is the advantage of bagging, because bagging is allowing or letting you to have the full control over the complete procedure to control the new species. Now, we will go back to the next question, and the next question is, what is the self-incompatibility, and why does the self-pollination not lead to the seed formation in self-incompatible species? So, this is the important question. Okay. So, there is a phenomena called self-incompatibility. What is this phenomena means is that even if there is a self-pollination happens in few of these species, it does not allow to have the does not allow the further growth of the pollen grains. So, what is the self-incompatibility? So, in few of the species of the plants, even if the pollen grains comes from the same plant through the self pollination, those pollen grains cannot be recognized by the stigma, and the stigma does not provide the proper environment for these for these pollen grains to extend its pollen tube and then deliver the male gametes for fertilizations. And this phenomena is known as the self-incompatibility. So, There are many reasons, and mostly it is governed completely by the chemicals which are being released by the anther and the chemical which are released by the stigma. So, what happen is, you can imagine that a pollen grain. So, pollen grain is containing a outer covering known as exine. So, a pollen grain is actually secreting the chemicals, and these chemicals are actually kind of attractant or kind of sensor machinery, and then you have a stigma of a flower, and this stigma has probably the receptors or probably another kind of chemicals. Okay? And these chemicals are important for this pollen grain to extend and grow and form a pollen tube, and this pollen tube is actually containing the male gametes. So, in this process, if any of these processes are going to be destroyed, because either the chemical which is secreted by the pollen grain or the chemical secreted by the stigma is not compatible with each other, this kind of plant is not going to control or not going to support the self pollination. Why this particular thing is important? Because it actually controls this actually controls the fertilization with another species. So, the self incompatibility could be possible with the within the species as well as with the another species, but the self incompatibility mechanism is involved to avoid the fertilization with another species, because you know that every plant species has different number of chromosomes. Okay? And if there is a one plant which contains the 32 chromosomes, and there is another plant which contains 37 chromosome or 38 chromosomes. This means this plant is going to have 16 chromosome in the female gametes, and this plant is going to have the 19. So, if this particular pollen grain will fall into this particular plant, and if this particular plant will allow them to grow, you are not going to have, you are going to have a very distorted hybrid structure and hybrid embryos, which probably will not give you a healthy plant. So, that is the one major mechanism, major reason why the plants have evolved a self incompatibility. And other issue is that the plants actually want to support the cross pollination, because they want to evolve into new species, and that is why they are sometime having the self incompatibility, means they will not get pollinated by the pollen grains coming from their own flower or their own tree, their own individual. This is happening at multiple stages. The self incompatibility could happen at multiple stages. What are these stages? The number one is the plant, as soon as the pollen grains fall onto the stigma, it may prevent the germination of the pollen tube. Okay. Number two, even if the pollen tube is generated, it will have the inability to penetrate stigma, because unless it will penetrate the stigma, it cannot deliver the male gametes for fertilization. Number three, it may not support the growth of the pollen tube. Number 4, well, even if the pollen tube has grown, it has penetrated the stigma, it may not reach to ovary. Number 5, even if it is reached to ovary, it may not allow the fusion of male and female gametes. So, and lastly, even if this process happened, that male and female got fused, the embryo may not be, embryo may get aborted. So, these are the multiple steps at which the cell incompatibility may take place, and the plant may not allow the fertilization, or plant may not allow the self pollination within the same individual, and this is actually a mechanism to let the plant to evolve into a newer and better species. So, after discussing these questions, we will continue our discussion about the sexual reproduction in flowering plant. So, what we have discussed so far, we have discussed about the flowers, its different parts, and then we have discussed about the development of male sex organs. So, once the male sex organs are developed, they actually start producing the pollen grains, and these pollen grains are being stored into the pollen sac. But pollen grains are not the actual sex organs or actual molecule which are actually participating into the downstream fertilization steps. So, pollen grains are actually being further matured to form the male gametophyte. So, today we are going to discuss about the maturation of male gametophyte. So, development of male gametophyte has two phases. The one phase is which starts, so in phase 1, which starts before the pre-pollination, means it happens while the pollen grains are stored in anther or pollen sac, and in phase 2 this happens after the pollination. Post pollination, when the pollen grains fall on stigma, okay, to give you the male gametophyte. So, let us see what are these developmental stages and how the male gametophyte is developed in the case of the angiospermic plants. So, as you can see on the PowerPoint slide, I have shown the different steps which are being taking place during the development of male gametophyte. So, as I said, there are two stages in phase 1, which are actually the pre-pollination stage, where the pollen grains are stored. This happens when the pollen grains are stored within the pollen sac. So, in the pre-pollination development, the microspore is the first cell of gametophytic generation, it starts germinating in situ, that is within the inside the pollen sac, and the freshly formed microspore has rich cytoplasm. So, as you can see, this is the microspore and it has a very large quantity of cytoplasm with a very prominent nucleus, and as it matures within the pollen sac, the nucleus which is present in the center then moved towards the periphery, and then it divides into two cells, and these two cells are formed by a unequal division, and these two cells are, one is called as the generative cells, which is this one, and the other one is called as the vegetative cell, which is actually this one. So, these because of this unequal division within the microspore, there are two different types of cells are formed. One is called as the generative cells, which is actually closer to the microspore's cell wall, and the other one which is actually within the center, and that is called as the vegetative cell. The generative cell attained ellipsoidal, lenticular or horseshoe shaped shape over the course of development, whereas and it has a limited number of organelles, whereas the vegetative cell has a lobed central nucleus, and the vegetative cell has a lobed central nucleus that lies in vacuolated cytoplasm packed with granules, starch, grains, lipids and proteins. So, these are actually the development happens when the in phase 1 during the pre-pollination stage. So, in the post-pollination stage, when the pollen grains falls onto the stigma of the female gametophyte, on the stigma the pollen grain absorbs the water and swells within a couple of minutes, and it releases the several factors which actually allows the recognition of this particular kind of pollen grains which is falling onto the stigma. And if you remember, in the past we were discussing about the restriction, how recognition system, how the pollen grains are being recognized, and how there is a self-incompatibility between to avoid the self-pollination. So, the factor which are being released from the pollen grains are allowing the pollen grain to germinate on the stigma or not, that is depend on the recognition system working between the pollen grain and the stigma. Subsequent to mature, once the pollen grain is being recognized that okay, yeah, this is the right pollen grain and it has to grow, then the pollen grain starts growing, taking the nutrition from the stigma. So, once the pollen grain starts growing, the vegetative cell enlarges and comes out through one of the aperture in the form of a pollen tube. So, if you remember, when we were discussing about the structure of microspore, we said that there are some projections from where the pollen grains actually are having the projections, and these projections can be used for growing the getting the pollen tube out. The pollen tube grows between the cells of the stigma and transmitted the tissue of the style. The wall and pollen tube is the wall of this pollen tube is actually the extension of the intine. So, if you remember, the microspore has two walls, one is called exine and the inner side is called intine. So, it is actually an extension of intine, and it grows, and during this process gradually the vegetative tube nucleus migrates to the pollen tube and reaches up to the distal end. So, while it is happening, the nucleus which is present into the pollen tube migrates towards the center or towards the end of the pollen tube, and these are actually and then the generative cell, which is actually this nucleus, then divide meiotically, and so then the generative cell divides to form the two male gametes. Each male gamete consists of a nucleus surrounded by a thin sheet of cytoplasm. It has been observed that a series of so this is what is happening. Once the pollen grain is fall onto the stigma, the pollen tubes comes out, and then once the pollen tube is extending, the generative cell, the vegetative cell nucleus migrates into the pollen and then divide to form the two nucleus, two male gametes. Each male gamete has a nucleus in the center surrounded by the cytoplasm, and now you can see that they are actually penetrating they are falling onto the stigma, and then they are penetrating the tissue of this particular the stigma tissues, and while they are moving, they are actually taking the nutrition from the stigma, and that is how they penetrate, and then these pollen tubes actually deliver the male gametes to the female gametophyte. So, with this we are done with the discussion about the male gametophyte, and now we will go back, we will discuss about the development, the other important flower part, and that is called as the female sex organ. So, in the female sex organ, which is also called as the carpel or gynoecium. It has three important parts. One is called stigma, and it is called as style, and it is called as the ovary. So, stigma is the actual platform which is on the top, and that is actually the area which is there to receive the pollen grains, and stigma is the only portion which allows the growth of the pollen grains and further maturation of the pollen grains to form the male gametophyte, and that is how when the pollen grains grow on the stigma, they give rise to the pollen tubes, and within the pollen tube the male gametophyte is developed, and that is actually delivers to the female gametophyte. Style is and the last part is the ovary. So, ovary is the female ovary is the female gametophyte, and it contains the many different parts, and now let us see how the ovary look like. So, this is what you see on the flower. We have three different parts: stigma, style, and ovary, and that is actually the forming the megasporium or the female sex organ. And within this we have already discussed about the stigma or the stigma and style. Now, we will discuss about the ovary. So, the ovary, a typical ovary contains the actually contains the female sex gametes or the egg, and that is actually the place where the seed or the fruit is developed. So, in a typical ovary, where it is actually a megasporangium in which the meiosis and megaspore formation take place. The ovule, which is the only place which is developed into the seed after the fertilization. The various part of the ovule and their function are as follows. So, as you can see this as a structure of ovule. So, what you can see, it has a long funicle, and it is like a stalk like structure. So, and which actually attach the ovule to the placenta, Then the next portion is the helium. Helium is the point of attachment of the body of ovule with the funicle, and then followed by the helium we have the raphe. Raphe is actually the longitudinal ridge formed by the lengthwise fusion of the funiculus with the body of ovule in a typical anatropous ovule. So, this is ovule, what you see is actually a anatropous ovule, and then the second portion is the nucellus. Nucellus is actually the area or the region where it is the mass of parenchymal tissue surrounded by the integuments. It encloses the embryo sac and provides nutrition to the developing embryo. Then we have the embryo sac, then this is the embryo sac. Then embryo sac is the female gametophyte which contains the egg apparatus as well as the other kind of molecules. Then we have the integuments. Integuments are the outer covering of the ovule which provides protection in the development of the embryo. Then we have a micropyle, and micropyle is nothing but the opening and it is a narrow pore or passage formed by the projection of the integuments through which the pollen tube enters into the ovule. And then last we have the chalaza, and chalaza, it is the place of origin of the integument or the basal swollen part of the nucleus. So, this is actually a typical structure of the ovule, but there are different types of ovule which are found in the angiospermic plants, and it depends on the orientation of the micropyle, means the orientation of the opening, and it is also been classified on the basis of the shape and the orientation of the ovule within the shape and orientation of the ovule. So, you can see that there are many types of ovules which are possible. So, these are the different types of ovules which are possible in the angiospermic plants. What you see is the A is called as the orthotropous ovule. In this ovule, the ovule is erect and it is devoid of any circumcurvature with the micropyle. So, you can see the opening is from the top and it is actually a straight ovule, and the longitudinal axis of the nucellus is parallel to the funicular axis. Next comes is the anatropous ovule. So, anatropous ovule is already we have discussed last time in the previous slide. So, in the anatropous ovule, the ovule is completely inverted in a orientation due to approximately 180 curvature of the funiculus. The longitudinal axis of the nucleus is parallel to the funicular axis. The example in the case of the anatropous ovule is helianthus, whereas in the case of the orthotropous ovule the example is polygonum. Now, it comes to the hemitropous. So, as the name suggests, it is actually the 50% rotation of the ovule. So, the body of ovule is twisted only halfway, so that the degree of curvature is intermediate between the orthotropous as well as the anatropous. So, it is actually in between the orthotropous and anatropous, and this means the micropyle is almost right angle to the funiculum. The micropyle chalaza line is horizontal and placed at 90 degree to the line of funiculum. The embryo sac is still be straight in all the three cases, and the example in this case is the ranunculus. Now, let us come to the next one, and that is called as the compylotropous ovule. In the case of compilotropous ovule, it is very different from the earlier three variations, that the body of the ovule is also getting twisted, and so that the body of ovule become curved, so that the micropyle and chalaza come to lie on each other side of the funicular. The embryo sac is more or less still be straight. So, there is no distortion within the structure of the embryo sac, and the example in this case is mustard. Then it comes to the amphitropous ovules. In the case of amphitropous ovule, it is slightly more distorted. The body of ovule shows prominent curvature and almost becomes horseshoe shaped. In this case, the embryo sac is also getting changed, and it is also get curved like the horseshoe shape. The funiculus is attached near the middle of the body. The micropyle, chalaza and helium come closer to each other, and the example in this case is called as is known as the capsula, and the last is known as the cercinotropous ovule. So, in the case of cercinotropous ovule, the ovule is getting twisted, and there is a long stack, or the long stack through which it is connected, and it is completely inverted, and the ovule is straight with micropyle facing upward. The funicule is elongated and appear as coiled structure. It completely encircle the ovule. In the beginning, the ovule is, so in this case, in the beginning the ovule is orthotropous, but during the course of development it get inverted due to the unilateral growth of funicule. So, as the funicular grows, it becomes further twisted. The example in this case is known as the obtunia. So, these are the different variations of ovule, and we will discuss further how the development of the female gametophyte is taking place. So, as we discussed for the male sex organ, the female gametophyte also developed in a very similar manner, that the first the few cells are being collected, and then they become what is the process of formation of female gametophyte? So, in the process of female gametophyte begins very early, when nucleus is not completely surrounded by the integuments. First of all, a single cell called the archaeosporium differentiate into the internal cell of nucleus near the micropylar end. The archaeosporial cells may directly function as megaspore mother cell, or it can further divide to form a inner sporangious cell. The sporangial cells or the primary sporangial cells behave like a mega mother cells or the megasporocytes. The diploid megaspore mother cell actually grows in size, and then it divides in size, divide by meiosis, so that the chromosome number getting reduced, and then it forms a classical tetrads. So, all these tetrads are actually containing the single cell, and these are containing the haploid nucleus. There are 4 cells in a typical tetrad. So, let us see from here how the female gametophyte is developed. So, the individual so out of the 4 megaspore mother cells, 3 megaspore mother cells goes for the degradation or degeneration, and these 3 mother cells actually provides the nutrition for the one of the megaspore, which is functional. Now, this megaspore actually goes for the 3 rounds of division. So, in one round it becomes 2 nucleus, in the second round it becomes 4 nucleus, and in the third round it becomes the 8 nucleus, 8 nucleated cell. Now, after this 3 successive mitotic division, it give rise to the 8 nucleated embryo sac. The developing embryos enlarge in size due to formation of the vacuole sacs in the center. Out of 4 nuclei, so out of these 4 nuclei, the 3 remain, 3 differentiate to produce egg apparatus consisting of an egg and the 2 synergids. Similarly, at the causal ends, the 2 remaining similarly at the causal end, 3 of the 4 nuclei differentiate as the antibodal cells. The 2 remaining nuclei, one at the micropolar end and one at the causal ends, termed as the polar nuclei, and they come into the center. They meet at the center and remain separate until the discharge of the male gamete or fuse to form the diploid secondary nucleus of central cell. So, this is the final embryo sac what you will see. So, in a fully developed embryo sac, you have the polar nuclei in the center, then you have the synergids on the micropolar sets ends, and then you have the antipodal cell on the causal ends. So, a fully developed new embryo consists of 7 cells, a single egg or the oospore on the microspiral end. It is polar in organization, in which the cytoplasmic elements are arranged towards the causal end and vacuolated cytoplasm towards the micropolar end. The wall of egg is common with the synergids. So, the wall is, so all these 3 cells are actually sharing a wall in between. After the fertilization, the egg develops its own cell wall and becomes the zygote. So, egg is the only active area which participating into the fertilization, and this egg is developing into the zygote. Then against to the egg, we have the 2 synergid cells. These are elongated cell which forms the filiform apparatus in the micropolar region. The cell wall is incomplete because it is sharing the few cells, cell wall along with the egg, and the filiform apparatus which is formed in the synergid cells helps in absorption as well as the transportation of the nutrients into the embryo sac. Along with that, the synergids also give the direction to the pollen tube by secreting some chemicals. So, that is how they attract the pollen tubes, so that it reaches to the egg. On the other hand, there are 3 antipodal cells, and they are vegetative cells of the embryo sac. They degenerate before or after fertilization, and their main job to help in providing the nutrition. Then there are single central cells. These single central cells, it contains 2 polar nuclei which fuse to form the diploid secondary nucleus. During fertilization, the 2 male gametophytes comes into the embryo sac. One male gamete fuses with the egg to form the zygote, and the other male gamete fuses with these central cell and to form a triploid nucleus, and that actually develops into the endosperms. So, with this we have completed our discussion about the flower, its different parts, and then we have discussed about the male sex organ as well as the female sex organ. We have also discussed how the pollen grains goes from the one flower to another flower, and at the end we have also discussed about the development of the male gametophyte as well as the female gametophyte. So, what we have got up to the now is that the male as well as the female gametophytes are evolved and developed. So, now in the subsequent lecture we are going to discuss about the fertilization and as well as how the pollen tube which is being coming out from the pollen grains are delivering the male gametophyte to the egg, and how the fertilization is taking place, and how the post fertilization the embryo sac is developing, how the embryo is developing into the embryo sac to form the seed, and how the seed is how the fruits are forming into these plants. With this, I would like to conclude our lecture here. I hope you might have enjoyed our lecture, and see you in the next class. Thank you.